this video, we are going to discuss about the further process of fertilization. Last class, we discussed about the fertilization process, that the fusion of gametes known as fertilization, and we discussed so many steps that how this fertilization process starts. As already I told that fertilization process is not as much that simple as we think. Fertilization is a complex process. Okay. So first of all, in fertilization process we discussed about the approach of the sperm towards the oviduct, because we know that fertilization process occurs at the ampulla isthmus junction. So after that we discussed so many other steps, what changes occurs in the ovum and when sperm's entry takes place, and what are the obstacles are there for the sperms and all. Now we will discuss that the sperm, when suppose this is the ovum, and sperm when enter or attached to the ovum site, which has many zona pedicellin layer and all. Suppose this is the outer layer they are having, is called a zona pellucida layer. Zona pellucida layer, and outer of that also there is a certain structures are there, which is called as corona radiata. Okay. So these structures are also there, corona radiata, and by utilizing the enzymes which are present in the sperm try to come towards the fertilizing site of the ovum. Okay, and after that, when sperms try to enter, at that time this ovum, exactly we know that what the ovum is not released from the oviduct. Suppose this is the ovary and it has the finger-like position that is called as fimbriae and infundibulum, and then after oviduct. And we know that the ovum released from the here towards the oviduct. But what happened here? We discussed about the oogenesis process. We already discussed what the changes occurs in the ovary. We can go through the last videos, that this ovum which has to be produced, which has to be give that fertilization process and zygote formation, which help to give the zygote and all, that is not completely divided. When the primary oocytes undergoes meiotic division to form the secondary oocyte, after the secondary oocyte has to form the ovum, okay, or you can say otites. This all has to be produced, but it is what in the secondary oocyte formation it get arrested at metaphase of the meiosis II. So in that way only it is released from the ovary towards the oviduct. So now this ovum which is there is not a completely ovum, right? So when this suppose it is now what it is, secondary oocyte, is not exactly the ovum. This is secondary oocytes. So this secondary oocyte get triggered by the sperm and undergoes further division to form what? Ovum. So it finally converts into ovum. The same thing will be that the ovum, it produces the ova, or you can say ova or egg cells, okay, and it also produces a tiny secondary polar body. Primary polar body is formed, takes place in secondary oocyte formation, and this is the secondary polar body which slowly going to degenerate, secondary polar body, and one ovum will produce. The same way again the zona pellucida layer is there, zona pellucida layers are also there, corona radiata also there, okay. almost all the things are the same, but the changes occurs that secondary oocyte has to convert into ovule, and it also has some of the vitelline membrane. As we discussed the structure of the ovule, that it has to produce what? Vitelline membrane, or the plasma membrane here it is called as vitelline membrane. Okay, so these structures are there in the newly formed ovule. After that, what happened? Sperm is there, ovum is there. Now fusion of the two gametes has to be takes place. So this fusion, where the fusion takes place? The fusion will take place in this region, ampulla and isthmus. In between these two regions, the fertilization process of the fusion of gametes takes place. So the sperms enter to this place, right? After that, see, suppose this is the fusion of gametes, you can say. This is the sperm and this is the ovule. Fuse together to form what? Zygote. How this fusion takes place? It is also not simple and single step. What happens in this? When the sperm ovum is ready for the fertilization process, here they will form a cone-like structure. Okay, a fertilizing cone will produce by the ova, and to that cone only one sperm can attach. Only one sperm can be attached to that fertilizing cone. After this attachment of the fertilizing cone, it enters. The tail part will not enter, but the middle piece and the nucleus will enter. How they can enter? Again here the acrosome is there. The acrosome upper membrane, which is there for their protection, will dissociate and they will dilute some of the enzymes. As already I told that corona radiata is disintegrated by this enzyme which is present in the lysine enzyme which is present in the sperm. Similarly, it will release the hydrolysis enzyme that also dissociate the or slowly disappear the corona. But before that also, what happened here? See, when fertilization cone attached to the ova, okay, it is just little bit upper side or towards the outside, so that the fusion of these sperms can be takes place. After that, what happened? Let's go with step by step. That corona radiata will not exactly dissociate here. Okay, after that it will dissociate or degenerate. Slowly it will get degenerate. And here what happened? The nucleus. Okay, suppose this is the ova, and outermost juna pellucidal layers are there, and corona radiata are there. It has what? Ectocentric nucleus is there, and the sperm nucleus also shared by the sperm. Now fusion of the gametes takes place. Fusion of gametes help to form what here? The fusion of gametes takes place, which is called as Syngamy process. Suppose, don't think about that, these are the changes. When fertilization cone is formed, the sperm enter and the fusion of gametes takes place. This is called as what? Syngamy process. As a result, what will form? Zygote. Okay, after this. Zygote is a single cell structure. So now what happened? This nucleus fused together to form what? Zygote. Suppose this is the zygote they will form, which is a 2n number, right? The number of chromosome is become 2n and single cell structure. After zygote formation, see again these corona radiates are there still now. Juna pellucidal layer is also there at these places. So after zygote formation, they immediately what process will start? After zygote formation, the next process is your cleavage. Okay, cleavage is a process by which the zygote immediately after the fertilization process or zygote formation process, the cleavage process will start. What is a cleavage process? It is a type of cell division. It is a rapid mitotic division. You can say this one as rapid mitotic division. Rapid. It is a rapid mitotic division. Okay. So we might have confused why we use that cleavage word instead of mitotic division, because it is type of mitotic division, but there is a simple or very what little changes are there in this mitotic division process and cleavage process. What are those changes, or what are the difference between the mitotic division and cleavage process? The first difference is, let us discuss differences. Difference from mitotic division. Why? How this cleavage process different from mitotic division? The first one is the daughter cells. In cell cycle chapter, we know that the daughter cells which are formed, suppose this is the cell cycle, this is the cell cycle, okay, there is certain phases are there, okay, that one G1 phase, S phase, G2 phase, it is M phase, okay. During cell division process of mitotic division, what will happen? It has certain growth phase, replication process, before replication process and after replication process. And there is a growth phase, means the size of the cell also increases. But in case of cleavage process, there is no chances of go through the growth phase. Means that, let me write here, no growth phase seen in daughter cells. Okay, there is no rapid cell division process, or you can say high rate of DNA replication process occurs. Rapid DNA replication process occurs. Okay, and third thing, high amount of oxygen also consumed during this process. High amount of oxygen also consumed in this process. And one more difference also we can notice that, see, there is no growth phase, means that, as we know that growth phase means there is a pre-preparation of DNA replication process. That means protein formation or lipid formation, all these things that takes place in the growth phase. So, and growth phase help to increase the size. But there is no growth phase, so there is no increase of the size. That means, you can write here, size remains same as in zygote. Means the division process will start, the single cell divide into a number of cells, but there is no enlargement of the cell takes place due to cleavage process. So it is a type of mitotic division, but from the mitotic division there are the certain points in what way they are different. So here what I told, that after fertilization process, immediately what will happen? Cleavage process will start, and this cleavage process help to form many daughter cells which going to form the new embryo, or you can say fertile life. How this will take place we will discuss in the next video.